Thank you so much for that very warm introduction. It's really an extraordinary honor to be here. As you mentioned, 2.5 years ago, I was an HHMI investigator in Memphis, Tennessee, and I was looking to expand the scope of our research, particularly to include electron microscopy and chemistry and cell biology. And I had this amazing opportunity to come here and join my colleagues and really be able to synergize with all of you. But as you all know, one of the challenges of moving to Munich is an issue of real estate. And what I could not have possibly understood when I had the opportunity to move here was I was offered primo real estate in Munich. My lab is next door to Matthias's. And it would have been impossible to predict how amazing having such awesome neighbors has been in transforming not just our science in terms of what we can do, but my vision as well. And so I just want to say a few words about this. When I moved here, I really did also want to incorporate mass spec into our work. And I laid out this condition, Matthias, I must have mass spec if I'm going to move here. But my view of this was based on reading the mind-blowing papers from the Mann department and from many of you. And I have to say my understanding of mass spectrometry was completely naive. I really didn't understand the power of mass spectrometry, and like Albert, I didn't understand the power of Matthias. So, unlike you, Albert, I'm not trying to dissect Matthias. I'm just trying to absorb Matthias and try to incorporate as many of these amazing features that you all intuitively understand into my own way of existence. So one thing that I've learned is that Matthias is light years ahead in his thinking. He asks, what is the important question, and not can we address that important question, but just breaks down barriers and starts to envision how to address an important question, even though no one has ever conceived of how to do this before. He's also an extraordinary mentor. He's populated the mass spec community and the scientific community with many of you because he's such an exceptional role model. And I have to say, you're not just a role model to your students, but it's been wonderful to be next door to you because you've become a great role model for me in terms of trying to establish vision and even how to try to address science. It's also not just science that he's extraordinary, and I think you also all appreciate Matthias's and way of living, really, his appreciation for the environment, his incorporation of technology into everyday living, and even his appreciation of music. So it's just been amazing. I'm just going to be able to touch the surface in terms of our science and where I hope to go with this. But I have to just thank you, not just for the privilege of the invitation to speak at the symposium, but for the privilege to be able to be your colleague and your friend. So thank you very much. I think we're all here to celebrate his second 30th birthday, and what's come along with this is the vision of clinical proteomics, where Matthias is really driving the front line to try to go from the clinic to use proteomics to understand mechanism, to use this as diagnostics, and to take this back into the clinic to be able to inform therapy, whether it's responses to therapy or to even design. I think this is a great example of Matthias's light year ahead vision, because while this might have seemed impossible a few years ago, I have a feeling that in a few years, everybody's going to think, oh yes, proteomics in the clinic is completely routine. And so part of the joy of being next door to Matthias is to try to also be able to come up with some way to incorporate this ourselves. So Matthias's department is called the Departments of Proteomics and Signal Transduction, and my department is called the Department of Molecular Machines and Signaling. I've left sort of the technique very vague because the common one-trick pony of my department is that we work on molecular machines, but we use biochemistry, structural biology, and in the move here we've also been able to incorporate some chemistry. And I hope that over the coming years we can play some role where we take the common part of our research, your interest in signaling and our interest in signaling, and be able to help contribute to your vision. Part of the way we have to do this is first to actually understand the vision and understand how to use mass spectrometry in the first place. And fortunately, in the first, since his last 30th birthday to now, We can also celebrate Matthias's vision of cell biology. And so some of our research has really taken full advantage of and built on Matthias's pioneering principles in cell biology. So I'm going to tell you, frame this talk first by telling you a little bit about me, because I am not a mass spectrometrist. But I'm going to say how mass spectrometry in the Mann department has shaped our thinking in the last 30 years, and give you two little vignettes about how we can go from cell biology through proteomics to molecular machines and signaling through structural biology and chemistry, and where we hope we'll be able to help Matthias in the future with his dream of using this, of going to the clinic. So around the time of Matthias's first 30th birthday, I started my PhD, and the view of, in the field was, at the time, was that the central dogma of biology is that one gene encodes for one protein, which encodes for one fold, and encodes for one function. And so I thought that the bottleneck in this would be really to understand how a protein adopts its fold, and how the fold then dictates its function. And so I spent many years as a PhD student studying protein folding with Peter Kim. But during this time, we realized there was a little bit of a problem with this idea, because due to the sequencing of the human genome, we realized that there are only 22,000 proteins by this definition of a protein, but then they have to do 1 million functions. So how does that happen? Well, Albert's already told us that this happens because protein form is dynamic, and while you see these protein forms as being dynamic by virtue of mass spectrometry, I think these protein forms are dynamics by virtue of them having millions of folds. But as we know that protein folds and function is massively expanded in many ways, one is by the formation of complexes. I set out to study this complex as a postdoc, where this is a kinase that's activated by this protein, but the whole complex is then inhibited by binding to another protein, and this achieves different folds and different functions. And we were very heavily influenced in our thinking about this through pioneering studies of mass spectrometry that were developed in the Mann department. Of course, we also understand that protein modifications vastly increase the number of forms. And again, our view of this has also been has been completely transformed by pioneering studies of the Mann department to identify acetylation using mass spectrometry. But during the time that I was a postdoc and studying this complex, I realized that there's another form of modification that I really fell in love with, and that is post-translational modification by another protein called ubiquitin. Ubiquitin here is drawn to scale, and the way ubiquitin functions is that its C-terminus becomes covalently linked to other proteins, which you then detect in your mass spectrometers. But for us, I was very interested in how this massive post-translational modification could change the fold and functions of its targets, and that's what we've been studying for the last 20 years. So ubiquitin is linked to proteins by a series of enzymes known as E1, E2, and E3. And that stands for enzyme 1, enzyme 2, and enzyme 3. I'm just going to briefly let you know that these enzymes perform a variety of interesting chemical reactions at the C-terminus of ubiquitin, so that ubiquitin at first is covalently linked to the E1 enzyme, then covalently linked to the E2 enzyme, and both of these are through very reactive bonds. And then E3s function in a variety of ways to promote ubiquitin ligation via an isopeptide bond that you guys detect by mass spectrometry to its targets. Now, ubiquitin can modify proteins in a variety of ways, but the most recognized function of ubiquitin is when polyubiquitin chains direct proteins for degradation by the proteasome. Oops. And this leads to massive regulation that we've been interested in for many years. Now, when I say massive regulation, I mean 5% of the DNA encoded proteome, and probably an even larger fraction of the number of proteoforms. Because in humans, there are 2 E1 enzymes that transfer ubiquitin to 30 different E2 enzymes that work with about 600 E3 enzymes in a variety of different classes that transfer ubiquitin to, in total, about 100,000 sites that you guys can detect by mass spectrometry on thousands of targets. And this is a vast regulatory mechanism that my lab has been trying to decode both by biochemistry and structural biology. So some of the things we try to do is we want to try to discover biological roles of ubiquitin by identifying pathways that regulate particular targets. And we're greatly aided with our collaboration with the Lamond department in this sort of discovery biology in this way. And we also want to understand the structural mechanisms of particular well-known important pathways. And so I'm going to just tell you two quick stories on how our knowledge of complexes and phosphorylation work together and how this has been influenced by mass spectrometry to mediate regulation, how mass spectrometry has influenced our studies to understand biological regulation, and then how other two modifications, acetylation by mass spectrometry, have allowed us to understand biological regulation. So my postdoctoral work was aimed at trying to understand molecular mechanisms underlying cell division. And even if you're not a biologist of cell division, which involves first alignment of chromosomes on the mitotic spindle and their separation into two cells. And this is really a marvelous hallmark biological event. But this event is actually very heavily regulated by ubiquitin. And I'll show you why in this movie. So this is a movie from the Gerlich and Peters lab. And I should mention our work on this has been a collaboration with Jan Michael Peters, who's the head of the IMP in Vienna, and Holger Stark, who is a director at the Max Planck Institute of Biophysical Chemistry in Göttingen. So you can really see here where the chromosomes are stained red, and another protein that regulates chromosome segregation is stained green. And what I'm going to show you is that this green protein has to be degraded for cells to divide. So if you look at any cells, after the chromosomes are aligned on the mitotic spindle, then the green protein is degraded and cells divide. And this is a very fundamental mechanism of cell division that we were very interested in. How is this green protein degraded? So the green protein is called securin, and actually this was partially discovered by Matthias and Wolfgang Zachariah here. And it is ubiquitinated by the E3 ligase anaphase promoting complex. Now another fundamental aspect of this regulation is that the timing of ubiquitination must be tightly controlled, or else there are catastrophic consequences. If the green protein is degraded when the chromosomes are not aligned on the mitotic spindle, then chromosome segregation is inaccurate. This can lead to aneuploidy, which is a hallmark of cancer. And so we wanted to understand this at a structural and biochemical mechanism. But first we had to understand what the complex was. So I was a postdoc studying the cell cycle when these papers came out, actually by our colleague Wolfgang Zachariah here, in collaboration with Matthias Mann, identified the subunits of the anaphase promoting complex, which is the E3 ligase that catalyzes ubiquitination of that green protein. As a structural biochemist, the way we wanted to work on it is to create the anaphase promoting complex to understand it, much like Albert is trying to recreate Matthias Mann to understand him. So the way we recreate things is that we use recombinant technology to make protein complexes in vitro. And the way people were making multi-protein complexes in vitro were that they would clone the vectors for the subunits of a complex identified by Matthias or yourselves. So the subunits of the anaphase promoting complex, you would make baculoviruses that express each one of these subunits, infect insect cells with 20 different baculoviruses, do an IP, and get an SDS gel that looks like pure protein. Now, we didn't have mass spectrometry at the time to analyze it, but what happens is that you actually get heterologous production of complexes here because not every cell is infected by every virus. To overcome this, we collaborated with Jan Michael Peters' lab in the development of a system where we can express up to 25 proteins at once from a single baculovirus. Every cell uptakes this virus, produces all 25 proteins at once, and we can make a homogeneous complex. And so this was very exciting. We could make the anaphase promoting complex, but unfortunately, we could not reconstitute the activity of mitotic proteins. That required something else. What was missing? Well, you can really see what was missing if you take an IP of one of the subunits of the anaphase promoting complex rendered on a silver stained gel from cells at this stage of the cell cycle, and you look at our recombinant anaphase promoting complex, and you can see that still several subunits are missing. We actually had those subunits in our baculoviruses. They simply didn't bind. So what was wrong? We weren't really studying the right proteoform of the anaphase promoting complex, as Albert says. So Renping Chow and Jan Michael Peters' lab, working with a mass spectrometrist at the IMP, identified 100 potential phosphorylation sites on the anaphase promoting complex, purified from mitotic cells. Now, this could, in principle, represent a problem for someone like me who wants to reconstitute a complex with 100 endogenous mitotic phosphorylation sites. But fortunately, it turns out that these were on phosphothreonine and phosphoserine, and these amino acids these modifications are somewhat similar to a naturally occurring amino acid, glutamate, that also has negative charges on it. So we were able to generate an anaphase promoting complex that had 100 glutamate mutations in it all at once, and actually be able to reconstitute the activity of the anaphase promoting complex. I'm not going to really show you the reconstitution of the activity, just to say that we were able to collaborate with Holger Stark in the determination of many structures of the anaphase promoting complex along the way. Oops, this is supposed to be running too. These are all dynamic themselves, and so we have many different conformations of this that allow us to understand now how this multi-protein complex, identified with the subunits identified by Matthias, and reconstituting modifications through pioneering methods, identified through pioneering methods such as those developed here, allow us to understand regulation. Now, this took us 18 years since Matthias identified the subunits working with Wolfgang in the anaphase promoting complex to determining the mechanism. And obviously, I don't even have 18 years left on my contract. So we really want to try to figure out how to accelerate things. We're still just learning how things work in the mind. And it's completely mind-blowing. I could never have conceptualized it, even if I wanted to in my wildest dreams. And so we're just getting this started, but I just want to say that we're really doing a good job, I think, of ramping this up. A year and a half ago, my lab couldn't even make proteins in insect cells, and we were just starting to even set up new systems. But we want to be able to use proteomics in a way to identify state-dependent changes that are dependent on ubiquitin, using mass spectrometry, and go all the way to mechanism. So less than a year and a half ago, Özge Karayel, sitting in the front row there, was able to identify 2 different forms of another E3 ligase that are involved in regulating metabolism. Shuai Chow in my lab has been able to, working with Rajan Prabhu, determine several cryo-EM structures that are morphed here. With the Mond department, we were able to, Jakob and Dawa in my lab were able to generate ubiquitination assays. I mapped the sites with Finn Hansen in the Mond department, and we started this a year and a half ago, and I'm happy to say that the first paper from my laboratory was greatly accelerated, also thanks to a collaboration with the Mond department that allows us to now ramp from proteomics to structure. I realize my time is already up. Do I have 5 minutes? To say how we can take this through the next way? All right. So we hope now that we've done this in 1 year, we've cut off more than an order of magnitude. I hope in the next 2 years we'll be able to cut off another order of magnitude in the speed to be able to learn how to be able to go from your identification of complexes and modifications to structure and mechanism, and the way you sort of are be able to drive biology forward. At least that's a dream of mine. Going forward, now, how do we advance from the clinics to proteomics? And I'm still trying to learn and conceptualize how to do this too, but I'll just tell you briefly about how proteomics has influenced our ability in one project to try to do this. We're not there yet, but at least we're trying. And this was work that, the clinical work here was started at Sloan Kettering. Mass spectrometry was carried out by Wade Harper, and then we've been doing the functional studies and chemistry with Kip Guy's lab. So Bhav Singh, who is a head and neck surgeon at Sloan Kettering, identified a protein he called squamous cell carcinoma-related oncogene 1, or DCON1D1, as being amplified and overexpressed in his patients with squamous cell carcinoma, and this correlates with mortality. So patients who have this amplification have, unfortunately, a worse prognosis. And Bub's lab showed that if they could silence this DCN1 protein in patient cells, then it preferentially caused apoptosis relative to cells that did not have this amplification. And so this seemed like a promising potential target for therapy. At least Bub has been trying to push this since he's been at Sloan Kettering. I should also mention that work outside of Bub's, just genomics data, has shown that this gene that we call DCN1 is amplified in a large fraction of squamous cell carcinoma patients. And that's because, it's amplified in a well-known amplicon. Now, we call this DCN1 for defective in cholinetylation because we've been studying cholinetylation for many years. What is cholinetylation? Well, NET8, which is the protein that's involved in netylation, is a ubiquitin-like protein. And even if you're a structural biologist, quite honestly, if you look at the structure of NET8 and ubiquitin, you cannot tell the difference. At least I cannot tell the difference after studying NET8 for 20 years. But unlike ubiquitin, which has thousands of targets, exists in many forms and in numerous functions, the best characterized function of NET8, which is 60% identical to ubiquitin, is really to modify 10 well-characterized targets that are subunits of E3 ubiquitin ligases. And so we've been interested in understanding how NET8 is put onto its targets and what NET8 does. Now, NET8 is basically a marker and a regulator of active E3 ligases. And NET8 activates about 200 different E3 ligases in humans. So NET8 basically netylated colons regulate the cell cycle, gene expression, development, numerous facets of neurobiology, and this pathway is also implicated in many diseases. Bacteria hijack this pathway, HIV hijacks this pathway, netylated colons are defective in many cancers, and fortunately there are now many therapeutics that are being developed to target netylated colons in sort of a blanket fashion. We wanted to understand how colon proteins are modified by NET8, and so we've been studying this DCN1 as squamous cell carcinoma-related oncogene protein for many years. And we were unable to reconstitute the E3 activity until work from Danny Scott using biochemistry in my lab and Eric Bennett and Wade Harper's lab realized that the E2 for NET8 is N-terminally acetylated. Now, those of you who do proteomics should be aware that about 80% of cytosolic proteins are N-terminally acetylated, and most people don't pay too much attention to this N-terminal acetylation. And so there really weren't actually knowledge of what N-terminal acetylation does. But Danny Scott in my lab realized that this N-terminal acetylation is required for DCN1 to bind to this E2. He solved the structure of this and showed that this N-acetylmethionine is buried in a pocket in this squamous cell carcinoma-related oncogene protein that is shown in surface. So there's the N-terminal acetyl binding in this pocket of this oncogene. We thought this could potentially be druggable, and we've actually went on a high-throughput screening campaign, identified a hit, we optimized the hit through medicinal chemistry, we made a covalent inhibitor, and we made a negative control that we cannot obviously solve the structure of. Our small molecules inhibit growth on soft agar of cancer cell lines that display amplification of this netylating oncogene. And because this talk is being recorded, I'm not going to show you the data, but we've actually been able to advance molecules that are now orally bioavailable and doing well in preclinical trials. So here we went from are interested in understanding the function and mechanism of ubiquitination and netylation to identifying this modification N-terminal acetylation by proteomics to be able to getting a lead that we hope will eventually be efficacious in the clinic. It's still a pipe dream, but at least we're understanding how to do this. So finally, I'll just close by trying to celebrate Matthias's vision of clinical proteomics. I hope that going forward, not only will the mass spectrometry and proteomics that you do completely change the way we think about science. It's just incredible vision, but we hope to also be able to use the techniques that we know how to do to be able to contribute back to help your vision really come true. And I'm just very lucky that I have a great team here. This is the group that's worked on the first paper from my lab in collaboration with Matthias. And yes, of course, location, location, location. What more can I say? It's great to be next to you, and it's great to be here. Thank you.